Hi everybody, this is Mrs. Mancuso and this is evolution video number 2. You'll notice on the screen in video 1 we focused on the areas to study in evolution and we focused on Darwin and natural selection. In this video we are going to look at what causes variety, what causes evolution, and how do you calculate Hardyweinberg problems. So let's get rolling here. Variety. We got to talk about variety. We know that it exists, and what are all the things that help give us a lot of variety. So the first thing that increases the amount of variety that we see is the existence of balanced polymorphism. So I know that you know poly means many, morphism means shapes or forms. So there's a balance in the environment between many different forms or phenotypes. So the fact that you can have tan rabbits and light colored rabbits and really dark colored rabbits, and they all tend to exist, that's an example of balanced polymorphism when you see many different types. Another great example is blood type. You could be A or B or AB or O, and there seems to be people with all of them. It doesn't seem to be a selective advantage. So when you see all these different varieties, that helps increase the chances of variety in a population. You could also think of it this way. Each of these things that we're talking about are what allows the variety to exist, and if it didn't exist, there wouldn't be as much, selection pressures might entirely wipe something out. But the fact that we have variety is pretty awesome. All right, next one, geographic variation. This means that you see differences in a population based on where they are living. They call this a cline. So there might be a northern and a southern cline of a population. That means they look very different based on where they live. A great example are rabbits. Rabbits that live in the north tend to be very white in color because they'll blend in with the snow, and they have shorter ears that helps hold the heat in better. And ones in the south tend to be more brown or muddled in fur color, and their ears are much longer, and that helps some of the heat escape in the months where it's warm. So differences in their phenotypes allow them to best survive in the environment that they're in. So here's our little rabbits, our little hares. All right, next one, sexual reproduction. This leads to variety in 3 different ways. So first of all, you have to remember that every time you're talking about sexual reproduction, you're talking about 2 parents. So you are constantly reshuffling and rearranging and introducing 2 different, 1 sperm, 1 egg from a different parent. So that is one way. You are getting a combination from 2 parents. So that's always leading to variety. But there are other things. First of all, independent assortment. That means that the chromosomes each lined up on their own before meiosis in the male and in the female. So there wasn't any lining up like all of, let's say this man, his sperm that's being produced didn't all come from the chromosomes that were his maternal ones from his mom, and they didn't all come from the paternal ones. It was a unique mix. That's what independent assortment means. The next one's random fertilization, and these aren't really in any sort of order. Random fertilization would come last. Random fertilization means you never know which sperm is going to fertilize that egg, and each sperm is unique and each egg is unique, so that is why siblings look different. There's always this unique combination, even though the 2 parents are the same. All right, crossing over. I wanted to have a little diagram here, so just as a refresher, here's chromosome 1 and here is chromosome 1. They're both replicated right now because they're about to go through meiosis. So on chromosome 1 here, I have this gene where there's a recessive N, and right here I have a gene where there's a recessive R. Over here, we'll say that the red stands for maternal chromosome. So let's say again that this is in a male and this is sperm that's being produced. The sperm that's being produced could have this N and R that is coming from this man's mom. Originally, they're his chromosomes now, or his chromosome that he originally got from his dad, which is a capital N and a capital R. And if those chromosomes lined up and there was no crossing over, you would see gametes that only had little n and little r or big N and big R. And that's it. No other combinations would occur because these genes are linked. They're on the same chromosome. So the way that we get variety in linked genes is through crossing over. So I drew synapsis here in the middle. Synapsis is the pairing up of homologous chromosomes during prophase 1 of meiosis. So when they crossed over, there was this little exchange. So if you look at my last diagram, here are the chromosomes that we were considering that they were from this man's mother originally. They came in the egg that first fused with the sperm to form that first zygote. So this was a maternal chromosome replicated. Now it doesn't look like a replicate anymore because there is a new piece of information on here. So now if you think about it, when this goes through meiosis, you could have a sperm that ends up with the little n and the little r. You could have a sperm that ends up with the big N and the little r. You could have a sperm that ends up with the little n and the big R. And you could have a sperm that ends up with the big N and the big R. So crossing over increased the variety. It was the only way that you can actually separate linked genes. So down here, I just wrote half of the gametes from this original one, if there was no crossing over. Half the gametes would have the homozygous recessive alleles. And over here, and half would have the two dominant alleles. But instead, when there's crossing over, you actually can have recombinants. So recombinants only occur once crossing over has happened for linked genes only. All right, next thing. This is our fourth type of variation, or fourth thing that causes variation, outbreeding. So I wanted you to first of all think, well, that's the opposite of inbreeding. Instead of becoming more and more similar, they're becoming more and more dissimilar, so the population's actually getting stronger in terms of its gene pool. So a great example are lions. A male lion will actually kick out his young sons. And when they are kicked out, they need to go and they need to find a new population of lions, and they have to be accepted and sort of win their place and be the king. So if they win their place and they are the king of that new area, they will be able to have the female mate with the females, and so then you are constantly outbreeding. So that actually allows for a great amount of variety within the lion population. All right, the next one here, diploidy. The fact that we actually have two of each chromosome is pretty awesome. It allows some of those little recessive alleles to hide, and especially if that lowercase, let's say it's a lowercase a, let's say it is something, that causes maybe a tan color, and maybe tan color doesn't blend in well and it would have been wiped out. If the environment changes and tan is favorable, we don't necessarily want to wipe it out of our gene pool. So it actually allows to stay in the gene pool the fact that you can be heterozygous and that little a could stay in your population. You have two of each. You don't express both of them, you just express one of them. All right, next one, heterozygous advantage. If it actually is beneficial to be heterozygous, big A, little a, then you are definitely going to keep that little a in the population. So a great example of this is sickle cell anemia in Africa. This doesn't happen here because we are not subjected to malaria, but in Africa, where water conditions are much poorer, they actually have a good chance of contracting malaria. But if they have the heterozygous form of sickle cell anemia, their red blood cells don't accept the malaria very well and they don't get sick with malaria. If they were little a, they would have died of anemia, meaning they wouldn't be able to get enough oxygen. If they were big A, they wouldn't die of anemia, but they might die of malaria. So it's actually an advantage in Africa to have the heterozygous condition, and that has allowed that little a to remain in the population. You'll find that with there are human enzymes that also have that same condition that occurs, that are active in cystic fibrosis and PKU and some other disorders. All right, our next section here. So we're done with C, we're on to D, the second of 3 parts of this video, causes of evolution. So what is going to actually cause change? Evolution is change in a population over time. There are 5 things that cause evolution. The first one is called genetic drift, and genetic drift is all about chance, and chance has a much greater effect when you have a small population size. So when we think about genetic drift, we're talking about a small population and how is its gene pool altering over time, maybe compared to what the original population was. So limited numbers, limited diversity. Chances are the allele frequencies of your small population aren't going to be the same as the original population. So there are 2 examples of genetic drift. The first is called bottleneck. So I actually want to get onto this picture first. So here I have the parent population with all of these genes, and a bottleneck means you're reducing the population size. So we have blue and yellow alleles. We'll say the blue are the dominant and the yellow are the recessive, and there's quite an even mix here. And when you pour them out, by chance, these survivors, these ones that are surviving this great reduction, so maybe there was a plague, or maybe there was Ebola, or there was something that actually really wiped out a lot of the members of the population. Looks like the majority that survived have the blue instead of the yellow, so by chance your population has a different gene frequency or allele frequency as the original population. This is happening right now in the cheetah population. They have been wiped out drastically, and the members that remain do not have the same frequency of their alleles as the original population. And now, because they are so inbred, there's such a small population remaining, their gene pool is very similar. Everyone tends to be quite similar, which leads to a lot of problems. Oh, you can also remember when we did the ecology unit, we talked about Isle Royale and the wolves and the moose, and the wolves were becoming so inbred that you were seeing a lot of problems. There was some spine issues and some leg issues that were in much greater frequency. I think they said 30% of the wolves had this disorder, whereas in a normal population it would be 3% or less. That's a good example of a bottleneck too, because that population is very small size. All right, next example of genetic drift is called founder effect. So if you take a part of a population and they leave, they go to a new place and they inhabit a new area, chances are their alleles aren't going to be in exactly the same frequency or percentage as your original population. So one really interesting one, I think, the Amish population has a much higher occurrence of polydactyly. Polydactyly, a dactyl is a digit or a finger, so polydactyly is having an extra digit, an extra finger, having 6 fingers or 6 toes. There's a greater frequency of that in that population than in the original Dutch population where they came from. So they also, I'm sure you realize, the Amish population, they do not go out and find new husbands and wives outside of their community. So there is a lot of inbreeding there, and their gene pool is a great example of founder effect. It's not the same as the original population. All right, our second thing that causes evolution is gene flow. When you have genes that are entering and leaving your population, you have change. So evolution is all about changing those allele frequencies over time. There we go. Next one, mutations. This is the first time this has actually come up in this unit, and it's really, really important. This is the only way that you can have a new allele. So new alleles are a random change in that sequence of DNA. So maybe it used to go A, T, 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 and instead it changes and goes A, T, C. So that altering of nucleotide sequence is the only way you're going to get a new allele. They're pretty random. We have a lot of things that actually prevent against mutations, our proofreading enzymes, the fact that DNA polymerase reads in a very specific order, but sometimes they do happen, and that is the raw material for evolutionary change. All right, fourth thing and fifth thing here that cause evolution. The fourth is non-random mating. The fact that organisms often choose their mates based on traits and behaviors, that's called sexual selection. So sexual selection is going to lead to evolution because it's choosing traits. And those that are chosen are the ones that survive and reproduce and pass on the traits that they have. So natural selection, our last one, we have already spent a lot of time on this. Of course, natural selection causes evolution. It's how the environment favors some phenotypes over other phenotypes. So you can think survival of the fittest. And when you see that difference, not just in surviving, but in being able to reproduce, remember it's all about the number of offspring that you have, that really is going to change your population. All right, now we got to talk about Hardyweinberg. So Hardyweinberg, 2 scientists, they worked on, hey, you know what? I think everything will remain status quo. We will have equilibrium in our gene pool. So that means the frequency of the alleles will not change as long as these 5 conditions are met. And you're going to have a lot of tricks that you're going to remember how these 5 things work. So here are the 5 conditions. Large population, no gene flow, no mutations, must be random mating, and no selection. And I'm hoping you're like, well, those are exactly the 5 that you just said. Yes, they are. So these are the 5 that you have to remember. If the 5 conditions that cause evolution are happening, you are not going to be in equilibrium. So to be in equilibrium, you can't have those 5 things. So here we go. We are going to talk about the math. there are calculations that you can do to determine the frequency of the alleles. So you are going to think of the letter P. A lowercase p represents the dominant allele. So we're going to talk about P's and Q's. Q's are going to be the frequency of the recessive allele. Ah, this is wrong. This word right here should be recessive. Let's fix it right now. This is what happens when I copy paste. There we go. Now it's right. All right, so P's are dominant alleles and Q's are recessive alleles. P's and Q's should add up to 1.0. So I'm going to add that right in here. P plus Q is equal to 1.0. So 100% of the alleles are P's or Q's. So this is very much a simplified process. There can't be other alleles like how blood type has multiple alleles. We just want P's and Q's, just dominants and recessives, and they should all add up to 100%. All right, now we have this other formula, and if you look at it and go, oh my goodness, I'm not going to remember this, it's on your AP Bio formula sheet, so you don't have to memorize it, although I'm pretty positive you will have it memorized within the week. So here's the formula. P squared plus 2PQ plus Q squared is also equal to 1. So 100% of the population is either this genotype, or this genotype. So if you think about it for a minute, last slide we were looking at allele frequencies, just the big A or just the little a. On this slide we're looking at genotypes. So big A is equal to P squared. Big A is equal to 2PQ. Little a is equal to Q squared. And the frequency of each of these genotypes, that's everybody. Everyone's got to fit in there somewhere, so that should add up to your entire gene pool. All right, last slide of the video. We are going to do an example. We will do plenty of these in class. This is a great thing to do as part of your Cornell notes. Make up some problems, or Google some problems, or go to your textbook and find some problems and solve for yourself. So we're going to solve this one together. I've got a population of 500 mice. 20 of those mice are white, and white is the recessive trait, so they have the phenotype white due to the genotype little a. So if I want to find out P, Q, P squared, 2PQ, and Q squared, I can find out everything from the information I have been given. I am given Q squared. So 20 out of 500 mice are little a. And I remember little a is Q squared. So the first thing I do here is say, I write down little a is equal to Q squared. And that is equal to 20 over 500, which is equal to 0.04. The next thing I do is I find the square root of 0.04, and that would be 0.2. So I have now found Q. The frequency of the recessive allele, little a, is 0.2. Well, the frequency of Q plus the frequency of P should add up to 1.0. So I can right away figure out what P is. P must be 0.8. So if P is 0.8 and Q is 0.2, that adds up to 100% of my alleles. Now, those were just the allele frequencies. I can also calculate the genotype frequencies. So Q squared is what I began with. Q squared was equal to 0.04. If I want that as a percent, 4% of the population is that recessive white. Next, I can figure out what percentage of the population is homozygous dominant, big A. So big A is equal to P squared, and P squared is 0.8 times 0.8, which is 0.64. So 64% of the population has the homozygous dominant genotype. The last genotype frequency would be the heterozygous. So big A is equal to 2PQ, which is equal to 2 times 0.8 times 0.2. Multiply across, you get 0.32, 32%. If I have done all my math correctly, it should all add up to 100%, which is the entire gene pool. Everybody must either be homozygous dominant, heterozygous, or homozygous recessive. I hope this has helped. Please review and do lots of practice problems. Ask me questions in class.